Welcome back to the Explainer. You know, right at this very moment, there is a literal microscopic puzzle flowing through your veins. Today, we're decoding the science behind blood groups and ABO typing. We're going to explore the underlying biology, map out our genetic traits, and actually step into the lab to see how this absolutely crucial test is performed.
But before we get to the lab, we have to establish why this matters so much. So, what actually happens if you receive the wrong blood type? This isn't just hypothetical trivia. The stakes here are incredibly high. Receiving blood from the wrong ABO group can genuinely be life-threatening. It sets off this massive internal alarm system that we really need to understand. So to figure out why a simple transfusion can go so drastically wrong, we need to zoom in.
Section 1. The microscopic world of antigens and antibodies.
Okay, let's dive into this. Our blood is a complex mix suspended in fluid plasma. And within this mix, your specific blood group is identified by antigens. These are protein molecules located right on the surface of your red blood cells. Think of these antigens as tiny, microscopic ID badges. They sit right on the outer edge of the cell, proudly displaying exactly what type of cell it is to the rest of the body.
Meanwhile, floating around in that fluid plasma, we have antibodies. These proteins basically act like your immune system's security guards. They're constantly on patrol, sweeping your system for foreign substances, germs or unidentified cells, and when they spot something that doesn't belong, they immediately alert the immune system to destroy it.
So, the absolute key here is grasping this distinction, because it's literally the foundation for everything we're talking about today. The antigens, those ID badges, are firmly attached to the surface of the red blood cells. The antibodies, our security guards, float freely in the liquid plasma surrounding those cells. The interplay between these two things is exactly what dictates your blood compatibility.
Section 2. The AB system matrix. Now, what's really interesting about this is how perfectly balanced the system is. Your specific combination of antigens and antibodies isn't random at all. It's a genetic inheritance that creates the ABO system matrix. If you have blood group A, your cells wear the A antigen badge, and your plasma contains anti-B antibodies. Group B is the exact opposite: B antigens and anti-A antibodies. Now, Group AB has both badges, so it has 0 attacking antibodies. It recognizes both as safe. And Group O? Well, it has no ID badges at all, so its plasma contains both anti-A and anti-B antibodies, fully ready to defend.
And this right here perfectly explains those life-or-death stakes. Just imagine someone with Group B blood receives Group A blood. The patient's plasma is already filled with anti-A security guards, so the second that Group A blood enters, it triggers an immediate, massive attack on those new cells. This is exactly why A and B blood must never, ever mix.
But hey, the puzzle doesn't stop at just 4 groups. We actually have to apply the rule of 8. There's another whole layer to our genetic inheritance that doubles our total number of blood groups from 4 straight to 8.
This brings us to the RH system. Remember how we talked about antigens as ID badges? Well, red blood cells sometimes carry an additional, entirely separate ID badge known as the RHD antigen. It's actually wonderfully simple. If this RHD protein is present on your red blood cells, your blood type is classified as positive. If the RHD protein is absent, your blood type is negative. That's it. That is the sole factor that dictates the plus or minus attached to your blood type.
So combining the 4 ABO groups with the positive or negative status of that RHD antigen gives us the definitive list of the 8 major blood types. Every single inherited possibility is covered: A, B, O, and AB, each with a positive and a negative variant.
Section 3. The lab and the ABO typing procedure.
Let's move on and see how this builds into real-world medical practice. How do doctors actually figure out which of those 8 groups you belong to? Well, we step into the lab and play medical detective. The ABO typing procedure is brilliantly simple. Step 1, take a blood sample. Step 2, mix that unknown sample in a tray with known antibodies against types A and B. And step 3, observe closely. We're looking for one very specific clue. Do the blood cells stick together?
Section 4. Interpreting clues and reaction results. So what does it actually mean when the blood changes right before our eyes? We're looking for a reaction called agglutination. When blood cells clump together after being mixed with our test drops, it proves a direct reaction just occurred. If we drop anti-A antibodies into the blood and it instantly clumps, we know definitively that A antigens are present. The security guards found their target. If it remains completely smooth, the antigen is absent.
Now this leads us to a seriously fascinating structural feature of Group O.
Because Group O red blood cells do not have any A or B antigens on their surface, there is literally nothing for the anti-A or anti-B antibodies to attack. If you mix Group O blood with any testing antibodies in the lab, it won't agglutinate, it won't stick together. In a patient's body, this means Group O blood can be safely given to any other ABO group without triggering that life-threatening alarm system. And this brilliantly illustrates what happens when we combine the O group with a negative RHD status. O-negative blood has absolutely no A antigens, no B antigens, and no RHD antigens. It is completely stripped of all surface ID badges. With no surface proteins to trigger an immune attack, O-negative blood is compatible with every single other ABO and RHD blood group out there.
Which leaves us with a critical real-world application. In a frantic medical emergency with an unknown patient, O-negative blood is the ultimate lifesaver because it essentially acts as a microscopic ghost, carrying no antigens to trigger an attack. But since only about 7% of the population has O-negative blood, it's always in extremely high demand. It really begs the question, as we unravel more about these microscopic antigens, how close are we to artificially stripping these proteins away to engineer a true universal blood supply right in the lab? Thanks for joining this explainer, keep questioning, and I'll catch you next time.